fourth class of lipid that we'll talk about are called the glycolipids. And again, its name tells us a little bit about it. The glyco part of it is telling us that there's a sugar present, which makes this the sugar fat. How could you go wrong? Notice that the glycolipids, I did not mention whether there was glycerol or sphingosine present, and that's because it could be either of those backbones. In the case of sphingosine, it will have a fatty acid and then the monosaccharide, which is our sugar. In the case of glycerol, it will have two fatty acids attached and the monosaccharide, which we call a sugar. And so generally, the monosaccharide can vary. Sometimes we actually have disaccharides or trisaccharides there, but there's at least one sugar present, and usually that sugar is going to be galactose. And yes, I do need you to remember that galactose is the sugar that we find commonly in the glycolipids. So here we have a galactocerebroside, and that means that it has galactose, which we see this monosaccharide, which is a 6-carbon sugar, and the sugar has OHs all over it. Notice all the oxygen. We'll talk more about that in our next unit about carbohydrates. But notice that it is a sugar attached to a sphingosine, and the linkage we see there is a type of ether linkage, not super easy to break. And then we're going to see next to that, we're going to see the fatty acid, which will be in the form of an amide. So these ones are fairly difficult to break apart, but they can still be hydrolyzed. And again, they have those fatty acids, and if there's a double bond in it, then it can go through the addition and oxidation reactions. And then to generalize this structure, we're going to see a head and 2 tails. So do we expect that to be found in a micelle, in a lipid bilayer, or in a fat globule? Well, hopefully you guessed right. It has a head and 2 tails, so we're going to find that in the lipid bilayer. And just as a reminder, sphingosine itself has its own long tail, and then the second thing that we'll have is a fatty acid for the second tail. In the case of glycerol, we've only got the 2 positions, so it's going to have 2 long tails. The third position is being taken up by the monosaccharide. There are 2 major types of glycolipids. They are the cerebrosides and the gangliosides. And you can probably think of what a cerebroside is if you think of what the cerebellum is. And yes, that means that these are found in the brain. And then the gangliosides are things that are found in the nerves. But they're very similar to each other. You don't need to know the difference between them, just that there are 2 different things, one called a cerebroside and the other a ganglioside. And again, that kind of gives us an idea of where we're going to find them in the body. These are found in the lipid membranes quite often because they have the 2 tails, but especially found in the brain and nervous tissue, in the cells that are in the brain and nervous tissue. And recall, this is where we said that the extra long tails are more likely to help us to protect the body from having anything pass through the lipid bilayer. And so, not surprisingly, oftentimes these have sphingosine. And when they don't, they have the 2 fatty acids, tend to be the longer chain fatty acids to help us with that blood-brain barrier. Let's take a quick detour to see how glycolipids determine your blood type. When we look at glycolipids, we find that the most common sugar that's attached to them is galactose. Now, galactose can have other sugars attached to it. We're going to look to see how do the different sugars that are attached to the galactose affect the blood type. In this case, we're going to see an antigen, and this is sort of a common baseline form that we see, and we see that there are 4 sugars here, so you can see these 4 sugars, and they're attached to some long chain. So this part is just our glyco part, and the rest over here will be your lipid part, which will be sticking into the cell membrane. But notice that we have this glyco part, and that's going to be sticking outside of the cell membrane, and that's where we have these antigens. Those are the parts that the body attacks if it's not the same, if it doesn't match what we have. So when we try to donate blood, one of the reasons they ask what is your blood type is if you give the wrong type of blood, there'll be something there that the body says, I don't know what that is, and attacks it. When you have type O blood, it has just this basic form. When you have type A blood, you can see that we've attached an N-acetylgalactosamine to the original galactose. What the body says is, I understand that if you're in a body that is A-type blood. But if you have B-type blood and the body sees that galactosamine sticking out of the end, it'll attack it because it says, that's not mine, I don't know what that is. Even though we do have galactosamine in the system, it's sticking in the wrong spot, and so the body attacks it. In B-type blood, we tend to have this extra galactose attached to the original galactose, and that is different enough that the body can tell the difference between them. So if you have B-type blood, you'll have this extra galactose, and if it has the galactosamine that we saw before, that would be attacked. So when a donor gives something with the wrong antigen, the body will attack it, and that is why we don't give the wrong blood type to people. So what do you think type AB blood has? You guessed it, it has both type A and type B antigens on it. And so we're going to see that it has both. Some of the glycolipids will have the extra N-acetylgalactosamine, and some of them will have just the extra galactose. So this type of blood is used to having both. That means that type AB can accept both A and B, but type AB cannot give to A or B because it will have the other antigen present and the person's body will attack it. Our body has something called an antibody, which is designed to attack anything that is foreign, things that it doesn't recognize. And this model for the blood type is going to look at these specific antibodies for things that attack the wrong blood type. So antibodies are part of our immune system. These happen to be Y-shaped proteins, so I'm drawing them as a Y. And then they're going to bind to any antigen, that's anything that does not match it, on the red blood cells. When they bind, it causes them to clump up and they'll destroy the cell, so they kill the cell when blood is placed in our body. If we put the wrong type in, it will be attacked by the antibodies, the cells will actually be destroyed, and it'll actually be worse than not having the blood. So, type A blood has extra galactosamine, type B blood has extra galactose, and type O blood doesn't have either, and of course type AB has both. Some of the glycolipids have added N-acetylgalactosamine, and some of them just have extra galactose. So we can look at this in a little more detail. If you have type A blood, your blood is used to seeing anything with type A in it, but will attack anything with type B in it, which means it can accept type A and type O. It cannot accept any B or AB. If we have type B blood, it's going to have anything that likes B, it will not attack. That means it will attack anything with A in it, so it cannot accept type A blood, but can accept B and O. It cannot accept AB because it has some A in it. Type O blood will attack anything with added A or B to it, so it can only accept type O. And finally, type AB will not attack anything because it's used to having both A and B present, so it is a universal acceptor. It can take A, it can take B, it can take O, and of course it can accept type AB. If type AB is the universal acceptor, it can take any type of blood. Which one is the universal donor? That's right. Type O is the universal donor because everybody can take type O because it doesn't have any added antigens. There's nothing for anything to attack, so everybody accepts type O. Because of genetics, of course, blood type distribution will vary depending upon where we're looking in the world, but on average, it's almost half of the population has type O, which means most people are able to donate blood, but not everybody can accept. The next largest one is the 42% with type A. Then we have about 8% which has type B, so it's fairly rare. And of course type AB is the rarest, with only 3% of the population on average having type AB blood.